Hi, my name is Tiffany Chen, and today I'll be presenting on yoga training's effects on inflammatory markers and cognitive function in women at high risk for Alzheimer's disease. Our study objective was to first investigate the inflammatory marker response to memory enhancement training, or MET, as compared to combined Kundalini yoga and Kirtan Kriya yoga. Second, we wanted to determine the predictive value of inflammatory markers on cognitive function through a randomized study targeting women with high risk for Alzheimer's disease, or AD. As an introduction, studies have shown that inflammatory responses can play a critical role in the neurodegenerative cascades of AD and mild cognitive impairment. Specifically, inflammatory markers such as tumor necrosis factor alpha, interleukin 6, kinase 3-like protein 1, interleukin 8, and C-reactive proteins are important signaling molecules in inflammation in AD. Previous research has shown that Kirtan Kriya increased telomerase activity, increased efficiency of brain metabolism, and decreased gene expression in the inflammatory pathways compared to relaxation in dementia caregivers with mild depressive symptoms. Our participants included women with current subjective memory complaints and high cardiovascular risk. They were randomized to a 12-week trial of the Behavior Intervention Memory Enhancement Training, or MET, versus yoga. Additionally, a panel of 17 cytokines were evaluated, and 3 factors were derived using factor analysis. Moreover, cognitive function was evaluated using the following measures, including learning, delayed recall, language, executive function, and processing speed. We also assessed for mood, perceived stress, and resilience. Lastly, regression models were used to examine whether cytokine levels are predictive of changes in cognitive domain scores. For our results, Figure 1 shows the average cytokine factor 1, 2, and 3 scores for participants in the memory enhancement training at baseline, 12 weeks, and 24 weeks. In comparison, Figure 2 shows the average cytokine factor scores for participants in yoga. At baselines, the group did not differ in any measurements. However, at follow-up, there was a borderline significant decrease of factor 3 and decreases in eotaxin and fibroblast growth factor 2 in the MET group, which can be linked to inflammatory properties. There was also significant decreases in epidermal growth factor and fibroblast growth factor 2 in the yoga group. Next, Figure 3 reflects the regression modeling graph of change in processing speed from baseline to 24 weeks versus average cytokine factor 1 scores for MET and yoga. We found that higher factor 1 scores at baseline was predictive of changes in processing speed in the MET group, but not in the yoga group at follow-up. Here, Figure 4 reflects the regression modeling graph in learning from baseline to 24 weeks versus average cytokine factor 2 scores for MET and yoga groups. We found that a higher factor 2 score at baseline was differentially predictive of change in processing speed in MET and yoga groups, but neither slopes were significant. Additionally, higher factor 2 scores at baseline was also differentially predictive of change in learning in the yoga group, but not in the MET group. In conclusion, there were no between or within group differences in changes in factor scores, but there were significant changes in individual cytokine levels for pro-inflammatory function. Initial predictive analysis showed correlations between cytokine factors and cognitive function outcome measures. Overall, inflammatory markers likely have multiple biological effects contributing to cognitive decline, and further study would determine the definitive role of individual cytokines. Additionally, reduction in inflammation and in the levels of circulating cytokines may serve as underlying mechanisms for explaining the efficacy of stress-reducing mind-body interventions on cognitive decline and Alzheimer's progression. I would like to thank Dr. Lavretsky's lab for their guidance and support. I would also like to thank Dr. Shaker Irwin and Dr. Noah Fetterman for their valuable feedback and continued mentorship within CTSI REP. Thank you for listening.